One day I decided to take my 5 dogs, Tuffy, Milo, Cooper, Lola, who is the biological parent of Cooper, and Rocky, for a walk in the park. While at the park, I found a picnic table, and so I decided to have my leftover taco that I had packed in my backpack for lunch. I walked over to the picnic table and placed my taco down on a paper plate. But before I started eating, I decided to take a picture of my most photogenic dog, Tuffy, for the gram. However, when I turned around, my taco was nowhere to be seen. No one messes with my tacos, and I was determined to figure out who was the taco thief. When I examined the paper plate, I saw that it was covered in dog saliva, so I knew that it was likely one of my dogs that must have ate it. Knowing that my dogs couldn't talk, and being a student majoring in forensic science, I knew how to take matters into my own hands. I took a saliva sample from all 5 of my dogs, including my witness Tuffy, and extracted a small sample of each of their DNA. I then used polymerase chain reaction, or PCR, to make many copies of their DNA at the location of interest. I then used a process called VNTR analysis. VNTR stands for Variable Number Tandem Repeats and uses the fact that a large part of our DNA contains a variable number of repeating DNA sequences. The image here shows a specific region of 2 homologous chromosomes from a diploid organism. Since the number of repeating sequences is unique to an individual, we can make DNA fingerprints using a process called gel electrophoresis. This is done by using special enzymes called restriction enzymes to cut the DNA and isolate the repeating sequences. Next, the DNA samples are loaded into a special gel that is permeable to DNA and given a positive charge at one end and a negative charge at the other. Since DNA is positively charged, it will want to travel from the positive end to the negative end. DNA segments that are smaller will travel farther than larger segments in a given amount of time as they can pass through the gel with less resistance, and so the DNA will separate in the gel based on their relative sizes. You can then compare the results with a DNA sample of an unknown individual to see if there is a match. So what I did was I loaded all the DNA samples into an agarose gel and performed gel electrophoresis. You can see from the results how Cooper and Lola have a DNA band that match. This is consistent with the fact that Lola is the biological mother of Cooper because Lola must have passed down one of her alleles to Cooper while the other allele must have come from the father. I then compared the suspect DNA to the evidence DNA. I was able to exclude Tuffy, Cooper, and Rocky as potential suspects who left the saliva at the crime scene because their DNA fingerprint did not match the DNA found at the crime scene. However, both Milo and Lola had DNA bands that matched the DNA at the crime scene at this particular location on DNA, and so I decided to perform another gel electrophoresis to see if I could narrow it down to only one suspect. So here I repeated the process for loci B. Again, we can see that the results are consistent with Lola being the mother of Cooper as they share one of their alleles. Now, if we compare the band patterns, we can see that the DNA from Milo does not match the DNA at the paper plate, and so we can eliminate Milo. The DNA from Lola and Rocky, on the other hand, matches the DNA from the paper plate, but since Rocky's DNA didn't match at the first location, then we can still exclude him, leaving Lola as the remaining suspect. However, that does not mean we can say for sure that Lola ate the taco. What it does tell us is that of the 4 suspects, Lola was the only one that had DNA that matched the DNA at the crime scene at the 2 loci that were examined.